The body relies on a highly efficient repair system to keep itself running. When you scrape your knee or grow an inch taller, your somatic cells divide through a quiet, predictable process called mitosis, yielding a perfect, identical copy every time. But that strict copy and paste routine only works for maintaining the organism you already have. When it comes to creating the next generation, those rules have to break. To create offspring, the body shifts to an entirely different cellular process known as meiosis. Its sole objective is to manufacture gametes, the sperm and egg cells. The production of gametes faces a strict structural constraint. A standard human cell holds 46 chromosomes. If a sperm and an egg each brought their full 46 chromosomes to fertilization, the math would instantly fail. Combining 46 and 46 yields 92 chromosomes. A cell overloaded with that much genetic material simply cannot function. For fertilization to succeed, incoming sets of DNA must be precisely halved to 23. The two half sets elegantly combine, restoring a stable genome. Without a mechanism to slice that genetic payload by exactly 50%, our biological blueprint would double and distort with every new fertilization. Complex life would collapse under the weight of its own DNA. This diagram details how a parent cell achieves exact reduction. Meiosis 1 starts with a diploid cell holding a full, duplicated set of chromosomes. The cell takes matching chromosomes from mother and father and binds them into pairs, visible during prophase 1. Here, a physical exchange occurs. The paired chromosomes cross over, breaking open to trade corresponding pieces of DNA. This acts as a massive genetic lottery. The cell drafts an entirely new, recombined sequence, ensuring no two gametes ever carry the exact same code. Once the exchange finishes, the shuffled chromosome pairs are pulled apart. The parent cell splits, separating the material into two distinct new cells. The chromosome count is actively being reduced, but the true accomplishment here is the variation. The raw material for unique physical traits has been forged before the cell has even finished its first split. Entering meiosis 2 on the lower flowchart, the two cells divide again without duplicating chromosomes. During anaphase 2, the sister chromatids are split to opposite ends. The process finalizes at the bottom row with 4 distinct cells, each holding a haploid half set of genetic instructions. Through 2 orchestrated sequential splits, a single parent cell transforms into 4 unique gametes, equipped with the precise fraction of DNA needed to create life. When you step back from the microscope, the effects of this cellular math become visible. It explains exactly why siblings sharing the same parents rarely look identical. Because of DNA reshuffling during crossing over, a child might inherit their father's eye shape while receiving a very specific genetic sequence that codes for their grandmother's nose. Every offspring receives a highly customized combination. If cells bypassed this step and reproduced identical copies, sexually reproducing populations would lack any meaningful variation. Entire species would exist as massive populations of clones, sharing the exact same genetic vulnerabilities. That variation creates resilience. Because individuals possess a diverse array of traits, a population always has members with the specific genetic makeup required to resist a novel disease or endure a harsh shift in climate. This allows complex life to root itself in almost every niche on the planet. By constantly rewriting the genetic code for the next generation, meiosis acts as the biological engine driving the survival of complex life on Earth.